Okay, good morning, everyone. So today we are reviewing for our first exam that's this week. Let's just go ahead and get right into it. So the exam is going to cover lectures 1 through 11, all the stuff that we've done so far in this first 4 weeks of the semester. So here we are in week 5. Today we are reviewing. On Wednesday is the first part of the exam. So the exam part 1 is multiple choice. This is done entirely over Laulima, right? So at 10:30 normal class time on Wednesday, if you log into Laulima and go under the tests and quizzes, you will see exam 1, multiple choice, become available. It will be available for until 11:30. Right? So you don't have to log into Zoom at all, right? In fact, there's no reason for you to log into Zoom. You just get into Laulima and take your exam. So when you log in and you see the exam, when you click begin, you will have 50 minutes to complete the 50 multiple choice questions. Right? So that means if you log in at 10:30, you don't have to rush and start right away, right? Because you have basically a 10-minute grace period. Just as long as you start the exam by 10:40, you will get the full 50 minutes before the exam closes at 11:30. All right. So log in, see the exam, don't freak out and start right away, read the instructions, and start when you're ready. Just as long as you start by 10:40, you will be fine. I made an announcement in Laulima with some more detailed explanations of how the multiple choice part works. So definitely go and read that so that there are no surprises. All right. Just a bit of advice. Because the multiple choice is set up so that you can only move forward through the questions, you cannot back up and review a previous question. Quick word of advice, don't rush through it at the beginning, right? And just randomly rush through it, because that's not good. But at the same time, don't get stuck on questions. So if you find yourself in the middle of the multiple choice part getting stuck on a question for, let's say, 2 minutes or something like that, you're better off just making your best educated guess, especially if you've narrowed it down to 2 choices, and just move on. Because the worst thing you can do is get stuck on a question, eat up a lot of time, still getting that question wrong. And then by the time you get to question 40, you have 2 minutes left, and you end up guessing on the last 10 questions that you could have gotten right, if you had paced yourself, right? So think about that. It's really not a good idea to rush through the last few questions because you end up spending too much time stuck on one early on. So you have to be able to commit and move on for the sake of giving yourself enough time to finish. All right. Then later this week on either Thursday or Friday in your recitation section, you go to recitation in person like normal, and that's where you'll take the free response written part of the exam. So your TI will proctor you. It's traditional paper exam where you write on the exam sheet. So it's going to be 10 free response short answer questions and worth 5 points each. All right. And make sure you bring a pen, right? Because your TI is not going to have a whole bunch of extra pens. Be prepared. All right. And then the last reminder is you do have your 4th homework assignment due this week as normal. And I know you might think that's a drag because you're supposed to be studying for an exam, but the point is the homework assignment helps you study for the exam, right? So do a good job on the homework, and I guarantee that will help you do well on the exam. All right. Okay. And I encourage you to try to get that homework done before you meet for recitation this week so that you can really benefit from having done the homework. Okay. All right, let's go ahead and start reviewing the material. So far for the first third of the semester, we have been getting a big introduction to the main macromolecule families, right, that form living cells. And we've begun to appreciate that we can approach molecular cell biology in different layers of complexity, right? Starting from the simplest, when we're just looking at how atoms interact with each other, forming bonds, and how it is that bonds represent electrons that are at certain sort of energy states, right? And some bonds are higher energy than other bonds, depending upon the nature of the atoms and how they're pulling on or being greedy for these electrons, right? And we learned how if you break a higher energy bond by putting energy in, enough energy to actually agitate the electron orbitals to the point where they literally, momentarily, right, get disrupted. If there are other bonds that are being broken at the same time with other molecules going through the same thing, then the electrons may rearrange and form lower energy bonds. And importantly, that difference between the higher energy bond and the lower energy bond is released as freed up energy that can be tapped into to drive biological pathways inside of the cells. We will study more about that later in the semester, but it's important early on to kind of understand that because all life is driven by chemistry. So from raw materials like water, carbon, small functional groups like amino groups, phosphate groups, and of course, oxygen, which is very important, they can come together to form what we consider small organic molecules, right? Molecules that are maybe made up of 10 to 20 atoms, so we might still consider them pretty small. Importantly, these small molecules are the basis of forming several of the very important large macromolecule families that our cells rely upon. Right? We learned about how 3 out of the 4 main macromolecule families are considered polymers. So polysaccharides, proteins, and nucleic acids are polymers because they're made up of long chains of monosaccharides or amino acids or nucleotides. And importantly, the fact that these long polymers are used in the cell is not by accident, right? Because long polymers have the advantage of being able to be broken down into the individual monomer units again, so that things can be reassembled, right? So that new polymer structures can be built. We said that lipids are the 4th macromolecule family that we cover in this class. Lipids are a bit different, right? Because lipids, right, are not made of repeating monomeric units. They are just long chains of carbons and hydrogens. So there's no real convenient way to say what an individual unit is. But lipids are just as important as the other 4 macromolecule families because these 4 components come together to form not only the structures of our cells, but also the machinery inside, right, that carries out certain functions, as well as information containing molecules, and also small molecules that can serve as signals. All right, so let's go ahead and review very quickly these macromolecule families. Let's start off with the carbohydrates. Remember, sugars are considered hydrates of carbon. So that name, if you keep that in mind, right, because carbohydrate, right, is a hydrate of carbon, hydrate refers to water. Right? It's not the same thing as hydrogenated carbon, right? Hydrogenated carbon is when you're adding individual protons, H pluses, to carbons. So for example, when we talked about lipids, right, you have saturated and unsaturated fats, right? That's when you talk about hydrogenation, right? But hydration is literally like the way we use the word hydrate in daily life. It has to do with water. We're putting water on carbons. So a simple sugar is a monosaccharide. That would be the individual monomeric unit. So we looked at glucose as perhaps one of the most important monosaccharides that our living cells use, both for energy and as the basis of forming large chains of polysaccharides. So glucose, the formula C6H12O6. Remember, you can either think of it visually represented in the linear form, so the Fisher kind of drawing, where you can see how the 6 carbons are arranged, and you can also appreciate where the oxygen on carbon 5 will tend to bind with the carbon 1, so that you end up essentially making this thing into a ring. And so on the right is the Haworth projection, or the ringed way of showing this, right? Either way, remember, we've been talking about how drawing things out with letters and sticks can be very misleading because it gives you a sense that this thing is this big sort of drawn out stringy looking thing, right? Or that it has like this really nice ring structure to it, when in reality, right, the real atoms that are involved and the bonds are not little sticks. The bonds are just shared or overlapping orbitals. So as a result, right, something that might look pretty complicated and big, like this ring here, right, in real life, the glucose molecule, if you just look at the amount of space it takes up, because remember, at the end of the day, that's all that matters, right? How much physical space these atoms are taking up and the shape of that physical space. It's actually quite a compact molecule. Recall that we have an alpha form of glucose and a beta form of glucose, all having to do with how the hydroxyl group on the one position, the carbon one, is either facing down in the plane or up in the plane, right? And how that can lend to the formation of alpha glycosidic bonds or beta glycosidic bonds. And why is that so important? Because once things are chained together, right, using either alpha or beta bonds, in order to get the monomers apart again and take the sugars off of the chain, you would have to have some other molecule that comes in and breaks those bonds. Right? And as we've been learning, those other molecules, most likely proteins, would be able to do their job only if the shape matches up exactly correct. The shape of the protein and the shape of the bond, this glycosidic bond that's going to be broken. So our bodies don't really have effective proteins that can recognize beta configurations of glucose chained together, for example, like in cellulose. So go back, review examples of monosaccharides, disaccharides, examples of long chains of glucose used in things like glycogen in our bodies, right, versus things like cellulose in plants, right, and how those different polysaccharides, serve different functions. Right? Right, moving on. Hydrocarbons. Hydrocarbons make up lipids. So again, hydrocarbons are just long chains of carbons and hydrogen. So lipids are hydrocarbon chains that have, for example, other groups at the end of them. In this case, a fatty acid would be a hydrocarbon chain with a carboxyl group at the end. So lipids are used for various functions like longer-term energy storage. They are a vital part of structural features like the membranes that make up our cells. And lipids, when they are free floating, can often serve as signaling molecules, as examples of other types of biological functions other than just energy storage and membrane structure. So there's a big diversity of different lipid structures, but all of them are more or less hydrophobic because of that long hydrocarbon chain. Remember, we discussed quite a bit the nature of water, and how water, because it's polar, any molecule that you dissolve in water or try to dissolve in water, as water being the solvent that's in our bodies, right? If that molecule that you put in there is not charged or itself is not polar, then the polar water molecules don't really have much interest in necessarily interacting with it. So they'll just keep bumping into it. Right? All these millions of water molecules surrounding, for example, a hydrocarbon chain, it's just going to keep bouncing against these carbons and hydrogens. And so that's why hydrophobic molecules end up just being shoved together eventually, right? That's why oil in water don't mix. The oil droplets come together. It's not so much that the oil loves hanging out with each other, right? It's more that the water has just all shoved them into one convenient spot, right? So review the 6 main classes of lipids. We've seen examples of fatty acids, triglycerides, phospholipids, glycolipids, be able to tell the difference between a phospholipid and a glycolipid. What are steroids, right? And what are terpenes, and examples of those from our lecture notes. Okay. Then we moved on to nucleic acids and the nucleotides that make up nucleic acids. So this is our structure of DNA. So you should review and be able to identify, recognize by sight the different nitrogenous bases Remember, we have 5 nitrogenous bases, 4 of which are used in DNA here. So adenine, thymine, cytosine, guanine. And then the 5th one, uracil, which is used in RNA. Right? So know those by sight. Be able to recognize the structure of an individual nucleotide. Right? So remember, a nitrogenous base, a ribose or deoxyribose sugar, and a phosphate group together makes a nucleotide. Right? In the DNA structure here, be familiar with the fact that it's the nucleotides or the phosphate groups that help form these phosphodiester bonds that make the backbone of the DNA strand. And they go from the C3 to C5 position, linking these together. Right? And be familiar with the nature of the hydrogen bonds that help hold the 2 strands of DNA together. Also, be able to explain why there's a 5' end of a DNA strand and a 3' end, right? And recognize that in a double-stranded DNA, right, the 2 strands are running anti-parallel. Okay. We discussed how DNA is replicated inside of our cells. We talked about how DNA along our chromosomes first begins to separate at origins of replication. And these are essentially bubbles that form, and the bubbles spread, right? As the bubble gets bigger, the 2 replication forks on either side of the bubble move in opposite directions, right? It's just like if you had a zipper, right? An old zipper, and you, instead of starting from the end, in the middle, you like pulled it apart. That's exactly what we're talking about here, right? The more you pull them apart, the bubble gets bigger, and the forks move in opposite directions. Forks being where the 2 strands are separating. So we said then DNA polymerase III, a large protein holoenzyme complex, big functional machine, can add free nucleotides to the growing 3' end of this copy DNA strand. Right? So the DNA polymerase is essentially scanning or reading along the template strand and using the DNA-based sequence of the template strand to serve, right, as the correct sequence to match up or pull in nucleotides on the left here to be complementary to it. And we said that if, for example, a thymine is pulled in here, it will stabilize with the adenine because of the hydrogen bonds, and it'll stabilize long enough for RNA Pol II to break these extra phosphates off of this DNTP, or deoxynucleoside triphosphate, using that liberated energy, those liberated electrons, right, in a high energy bond that's broken, to be able to drive reformation in a lower energy bond, in this case, attaching the oxygen from the phosphate group, right, to interact with the hydroxyl group coming off of the neighboring or previous nucleotide that was added there. And remember, DNA Pol III can only make the new strand in a 5' to 3' direction. Okay. So in addition to DNA Pol III, there were other proteins involved, because as the zipper is being separated, right, or as this replication bubble is now forming a replicon that is spreading, recall that on one side, right, the template strand, or the navy blue strand, can be read very easily by the DNA Pol III. In this case, the DNA Pol III is going from the right side to the left side. Here's the DNA Pol III, and it happily makes the leading strand as one continuous strand that basically keeps following the separation, or the fork. As the fork keeps spreading to the left, this DNA Pol III complex can just keep cruising along. But we learned that the problem is the other template strand. The other template strand is in the opposite anti-parallel orientation. So in order for DNA Pol III to copy that, it can only do it as little fragments, because as the zipper opens up, right, another DNA Pol III would have the opportunity to get in here. But of course, the zipper keeps moving to the left. The DNA Pol III on this strand can only make a new strand heading off to the right. So that's why this lagging strand is made of first putting small random RNA primers onto the DNA. So a big primase complex takes care of that. Then the DNA Pol 3 can elongate the primer, because remember, DNA Pol 3 cannot start from scratch. It has to have an existing fragment of DNA with a free 3' end on it in order for the Pol 3 to start adding things to it. Okay? And then eventually the Pol 3, the light blue lagging strand, it'll bump into the next spot where a primer was put down. Then you'll have other proteins go to work. DNA Pol 1 will remove that red RNA primer, because you cannot have RNA left over as part of a DNA molecule, right? Because its ribose sugar is not the right shape, right? Compared to the deoxyribose sugar that's involved in the DNA. You would not get the normal DNA double helix if you had these weird little glitches along the way because the bond angles aren't correct, right? And then you have DNA ligase, this big blue enzyme complex that links these Okazaki fragments together. All right. So then once we learned about those main components, right, we then started talking about the central dogma of cell biology. And that is that information flows from DNA through intermediate RNA molecules, right, into finished products like proteins. Okay? So be familiar with what the central dogma of cell biology is trying to convey. Right? And I want you to notice something, even though I don't think I very clearly stated this previously, hopefully you realize, right? This is all about genes coding for intermediate RNA molecules that then ultimately are involved in coding for proteins, right? So where are the carbohydrates here? Where are the lipids, right? Where is it that the DNA being made, that process, where does that fit in, right? Those are all the roles of different proteins. Right? So in other words, our genes do not code for carbohydrates. Our genes do not code for lipids, right? And in many ways, if you think about it, the functional purpose of a gene is not to code for another DNA molecule, even though, of course, it serves as the template when DNA is being copied, right? The purpose of genes is to make proteins. And the functional proteins are what take care of putting together carbohydrates that come from our diet or come from other being recycled stuff in our cells, or putting together lipids, right, that come from our diet or from other lipids that are being recycled in the cells, right? So carbohydrates and lipids, they are not things that are actually coded for by our genome. Right? Our genome codes for the proteins that function as the little machines that take care of putting together those other important macromolecule groups. Okay. So go back, review. What is a messenger RNA? What is a tRNA? What is an rRNA? Right? Why is it that even though the messenger RNA is the actual coding sequence for an eventual protein, why is it that tRNAs and rRNAs are essential, right? Still essential for proteins to be made in the first place? So we began to learn about how our DNA is accessed inside of our cells, right? How the multitude of different sequences on the different chromosomes of our DNA are controlled in terms of whether our cell has access to that information, the DNA base sequence that represents a gene. We said that the DNA in our cells is not naked, free-floating DNA. It's tightly compacted due to its associations with different types of proteins that help pack it up into a complicated structure that we don't just call DNA anymore, right? Because it's DNA plus all kinds of proteins. We call that chromatin. Right? And we said that one of the most fundamental parts of chromatin is the fact that along the length of a chromosome, you would have about 146 base pairs of DNA wrapped around a nucleosome made up of an octamer, or 8 individual histone proteins, forming this like bead, right? So along the DNA strand, you'd have many of these beads, so it looks like a string of pearls or a beaded string. Okay? We said that importantly, the tighter that these histone proteins hang on to the DNA, the more compact the overall chromatin would be, making it harder for the genetic sequence in that DNA to be accessed or read. Right, so we would say that would be heterochromatin, versus if the histones loosen their grip on the DNA, and let's say even to some extremes, the whole nucleosome falls apart, the histones fall apart, right? Then that section of DNA becomes available. It becomes more like euchromatin, chromatin that's opened up, and it is more transcriptionally available. How does that happen? We learned that different types of modifications on the tails of histones, things like acetylation or methylation, right, alter the shape of the histones and influence how tightly the nucleosome hangs on to the DNA. Again, you do not have to memorize this histone code, but you do have to understand why it's so important that the modifications of histones done in this fashion, right, is an important mechanism by which our cells are dynamically using different stretches of your chromosomes. Right? Okay. So we said that if you have a region of a chromosome that is sort of unraveled, so more like euchromatin, so that other proteins and things can actually bind, get in there and see sequences on the DNA, along that stretch of the loosened up DNA, you might find several sequences that actually represent genes, right? Separate sets of instructions for making some other molecule. Okay? We learned that if this is a stretch that's actually the coding the body of a gene that's being transcribed here, okay, you would normally find just upstream of it some sort of a promoter region, right? And this region consists of highly conserved short DNA sequences that make the DNA, or the actual shape of the DNA as the DNA is curving around, like its surface features, right, would be the right shape for particular specific proteins to come and sit on the DNA. Oops, sorry. So we said, for example, in this picture here, you imagine you're looking at this promoter region, and in the DNA, there's a short green region, red region, et cetera. These sequences, right, would make the DNA's surface just the right shape for particular proteins known as transcription factors, like this green protein here, to fit right in there. And as these different transcription factors are binding to these different short sequences in the promoter, right, they pile up, and their overall shape, right, the big blob that they end up forming, is the right shape that the RNA polymerase II complex will then dock to, right? Because it's the right fit for RNA pol II to sit on there and be lined up right at the transcription start site for this gene. And then down here, right, this big golden blob, which is the RNA pol II, it was lined up by the promoter, and it starts transcribing, right, transcribing a primary RNA transcript from this gene. So remember that our genetic code is coded for by every 3 DNA bases, ultimately, right, representing, for example, the identity of one of 20 amino acids that should be put in the finished protein. So remember that proteins are chains of amino acids, right, and the order of amino acids, you have 20 to choose from, right? The particular order of amino acids will end up determining the shape that this big polypeptide chain collapses into. Right? And that shape then has to be the right shape in order for it to interact with other molecules, whether it's other proteins or the DNA, right, that has the matching shape. So we said that in order to be able to specify which of 20 amino acids goes in the next spot on a protein, we have to use, or life uses, 3 bases. Because 3 bases, if you have 4 choices of which base in each position, 4 times 4 times 4 gives you 64 possible amino acids. We only need 20, right? Because billions of years ago, life figured out that it could just get away with 20 amino acids, right? So we have a lot of extra leftover triplet codons, right? So that's why our genetic code is redundant. So when you're given the big table of triplet codons, right, there's far more than just 20 triplet codons. But I think by now, right, you're familiar that many of those codons code for the same amino acid. So this is a nice reminder that in this figure here, right, the peach colored molecule is the resulting messenger RNA that was made by RNA Pol II reading this section of DNA over here. Remember that at first the RNA Pol II makes a primary RNA transcript, right, or a pre-mRNA that then has to undergo editing. Right? So introns are removed, exons are kept in the RNA strand, resulting in several different possible variations that represent the finished messenger RNA that is then moved out of the nucleus into the cytoplasm where it can be translated, right? So go back, review what we talked about as to why life evolved to use mRNAs or RNAs in general, I guess, as an intermediate molecule as part of the central dogma. Right? What advantages that gives us in terms of flexibility. If you have one gene in the DNA, right? What does having different possible edited versions of RNA give you? Okay? So you'll be responsible for being able to translate, for example, a messenger RNA into the finished amino acid residue sequence based upon being given that degenerate code table. Right? Okay, so recall that the translation that happens of a finished mRNA in the cytoplasm being translated into the finished protein involves all three types of RNA molecules that we looked at. Obviously, the mRNA is the fully edited sequence that's going to give you the codons, right, that specify what amino acids should be strung together. Where does the stringing together of those amino acids take place? In the ribosome. Right? And so the ribosome then will read the mRNA, right, and these tRNA molecules will be coming into the ribosome, each tRNA bringing a specific amino acid with it. Right? And on the anticodon loop of the tRNA, right, you have these three bases that should line up. Once the tRNA gets into the ribosome, it lines up with the codon and the mRNA. And if it matches, then that tRNA is stabilized there. And whatever amino acid it's carrying on its other end, right, now can be incorporated as part of the growing polypeptide chain of amino acids. Be familiar with why we say the third position is the Wobble position, right? Understand how the Wobble position works, okay, in terms of allowing for our genetic code to be degenerate. In other words, allowing for multiple codons in the mRNA, like you see on the left here, right, to code for that one particular amino acid. Okay, so then we began to look at the anatomy of a ribosome. So we said the ribosome is a massive structure that's made of multiple protein units, as well as ribosomal RNAs, right, or rRNAs that are all sort of coiled up in here. And together, right, this combination of proteins and rRNAs is like a big factory, right, a big factory that produces proteins. So remember that the whole ADS ribosome is made up of these two halves, the 60S and the 40S. There's a channel represented by the dotted line through here that the ribosome is actually going to scan an mRNA that's being fed through it. And we learned that inside the ribosome, there are these three chambers or pockets, right, called the A, P, and E. If we look at this figure over here, right, we can see that tRNAs that come in, right, dock in the A site. Right? The previous tRNA that was in there is moved over to the P site. That previous tRNA is hanging onto the growing polypeptide chain that is now emerging from the ribosome. Importantly, if this new tRNA that came into the A site properly docks on the mRNA because its anticodon matches up with the next codon sequence of the mRNA, it'll be stabilized there. And then the growing polypeptide chain will be moved over from the tRNA in the P position that was hanging onto it. It'll be put on top of the amino acid here that was brought in by the new tRNA. And therefore, everything moves over to the left. This tRNA would move into the E site and then leave the ribosome. And then this new tRNA in the A site will move over into the P site. And now it is hanging on to the polypeptide chain with one more additional amino acid that just got added on. Okay. So go back, review the steps of translation that involves initiation, the ribosome coming together on the mRNA. Keep in mind, right, it's really important on the top of this figure here, this figure represents a finished edited mRNA molecule or messenger RNA that was edited in the nucleus. It's now out in the cytoplasm. It contains on it features such as, obviously, the middle dark orange part is the coding sequence for whatever protein this is eventually going to be translated into, but there are other parts of this mRNA that never get translated. Right? So there's a 5' cap on one end, a poly A tail on the other end that serve to protect this messenger RNA molecule from being degraded by the cell. Why? Because remember, I reminded you that oftentimes viruses carry small RNA genomes. And if they infect your cell, these free-floating RNA genomes might end up being integrated into your cell's genome and hijack your cell and make your cell make more virus particles, right? So way back at the beginning of life, it was important that cells develop mechanisms to recognize that they might have viral genomes in them, and they should get rid of them. So that means early on, right, our cells evolved mechanisms for not liking free-floating small little mRNAs. So our cells, of course, have to make small mRNAs as part of making proteins. So the solution was to put modifications like a cap or a poly A tail that help protect this RNA from being degraded, so that your cell will realize that this is actually an mRNA that the cell itself made. Right? And also those caps and tails help provide the way that the mRNA is moved from the nucleus out into the cytoplasm, because different proteins can attach to those and move them out. So then you would have a start codon in green here. Remember, that start codon is the codon AUG for methionine. So as the ribosome comes together, it comes together on the start codon. The first tRNA is a tRNA that carries methionine, so amino acid 1, okay? And it's going to start off in the P position. Elongation involves additional tRNAs being brought in by elongation factors. And we said that if the anticodon, if the next tRNA does match, then it'll be stabilized and the growing chain of polypeptides gets moved over. Right? If it doesn't match, then no big deal. The tRNA would just fall out, right? Because it would not be carrying the right amino acid that's coded for by the next codon in the mRNA. And then eventually, as the ribosome or big line of ribosomes, remember, because it's a whole chain of ribosomes, as they're reading down the mRNA, it's going to hit the stop codon, right? And then proteins would then encourage the polypeptide chain to be released. And then these would go off and fold up into your finished protein. Okay. So speaking of proteins, let's go ahead and review protein structure, right? Because we looked at we already reviewed the other 3 macromolecule classes as part of today's review. Let's go ahead and review the 4th one. Right? So as the mRNA is being scanned by the ribosome, right, and this growing polypeptide chain is being put together, as a reminder, right, it's made up of individual amino acids, 20 amino acids, each of which has the same sort of central structure of a carbon with a carboxyl group, an amine group, a hydrogen sticking off of it, and then this other variable side chain group. Right? And I said that, yeah, in real life, in the universe, there's an infinite number of different small little side chain groups attached to it. Even surrounding us every day on Earth, there are different variations of amino acids that would fit this. But the important thing is life only needs 20 of them, right? The 20 that life learned to get away with using billions of years ago, right? So go back and review how it is that these individual monomers, these amino acids, are put together through condensation reaction, right, to form peptide bonds. And so how a big chain of these amino acid residues, right, is a polypeptide chain. That's our primary structure, the sequence of amino acids. Then the secondary structure is how those, the central part of all the amino acids being the same themselves already just have some interaction and different bond angles, right? And then the tertiary structure is how the side chains of all these different amino acids, whether it's this bulky valine side chain or this bulky isoleucine side chain or this tiny small methyl side chain, right? The order of amino acid means all these side chains have to deal with each other because they're getting each other's way, how that will then influence the polypeptide chain to fold in very particular orientations to make your finished protein. And that's your tertiary structure. So recall that as you have a line of ribosomes that have begun to go to work translating an mRNA, as the growing polypeptide chain starts coming out, right, many proteins, the chain of polypeptides that come out will just fold up in the right way that that protein is meant to fold up. So that means that these proteins can just be made right into the cytoplasm of the cell, and then they can be moved to other parts of the cell where they're needed. Right? So for example, certain proteins like histone proteins or other proteins that are used in the nucleus, right, are just made in the cytoplasm like this and then moved into the nucleus. But many other proteins have to finish their translation in the endoplasmic reticulum. Right? So in this case, you have many proteins that as the polypeptide chain comes out of the ribosome, translation is halted so that the whole mRNA with the ribosomes attached to it is brought to the ER. And we learned that one of the reasons why that's important is because as you have a polypeptide chain growing, right, those residues that are coming out might already start folding up with each other because of their side chains. And that might not actually be the resulting shape that you want, right? Because maybe the actual shape of the protein you want is dependent upon the rest of the polypeptide chain being made so that then whole thing can fold up properly, right? So that's one reason why co-translational import of these proteins is done into the ER. And in the ER, you would have other proteins like chaperone proteins. What's a chaperone? It's just like a field trip for school, right? You have the parents who go along with the third graders. They are the chaperones, right? To make sure that the kids in the school bus behave properly, right? So you have chaperone proteins in the ER that help fold the polypeptide chain in the proper way it's meant to fold. Another important reason that you do this co-translation is many proteins are meant to be embedded in cell membranes. So we learned how certain sequences of amino acids, as they're coming out from the ribosome, right, might make the polypeptide chain get stuck in the membrane. And depending upon how they get stuck, they end up forming proteins that are embedded in that part of the membrane of the ER. And that part of the membrane of the ER can then bud off as part of a vesicle. And then that vesicle can move off somewhere else. For example, to the outer boundaries of the cell, where that vesicle will fuse with the cell membrane. Because again, the membrane of the ER is the same kind of membrane as the outer cell membrane, right? And then those transmembrane proteins would then be introduced to the outer cell membrane where they'll be stuck there. Okay. We also said that many proteins, when they achieve their final shape, maybe they're meant to be machines that need to be turned on or turned off when you need them to, right? And how do you turn on or turn off a protein? Well, remember, in order for a protein to work, it's all about three-dimensional shape. Right? Either it's the right shape to interact with some other molecule, or it's not the right shape. So we said that post-translational modifications to proteins are a very common way in our cell to control the on and off status of certain proteins, as well as labeling certain proteins. For example, if a protein needs to be recycled, oftentimes marks can be put on them. So these little chemical modifications include things like adding methyl groups, acetyl groups, maybe even phosphate groups. Right? Those of you who think about what we discussed with the histone tail modifications, remember the histones that make chromatin and how the N-terminal tails are sticking out and it can be acetylated or methylated. I mean, that is exactly a perfect example of post-translational modifications of a protein, right? In that case, the proteins are the histone proteins, and they're being post-translationally modified through adding methyl groups, acetyl groups, right, to cause the histones to behave a certain way. right? But this type of post-translational modifications can be done on many different types of proteins. So recall that adding something like a bulky phosphate group, so here we had a serine amino acid residue somewhere buried inside this big polypeptide chain, right? Remember, if you were to unravel this big protein, it could just be one long polypeptide chain like the one up in the cartoon over here. And so we're saying at some point, one of these amino acids, right, is the target of being phosphorylated. In the finished tertiary structure, that amino acid might be buried somewhere in here, the way that the chain folds up. But when it gets phosphorylated, right, all the other side chains of its neighboring amino acids, right, they have to put up with this. They have to deal with it. They have to adjust because the shape of the side chain is different now. So all the other side chains that are neighboring it have to kind of shift around a bit to get comfortable again, right? Because all of a sudden there's this change in the shape and there's a ripple effect. There's a ripple effect that then might cause the overall shape of the protein to reveal these brand new three-dimensional features like pockets or clefts that might be the interaction shape, right? Or the interaction site where some other protein now fits in there or some other molecule fits in there. Right? So this post-translational modification of proteins is a very important part of protein function, but we call it post-translational because when does it happen? It happened after the protein finished being translated and is now floating around somewhere, right, waiting to do its job. Okay, so that's it for the review today. I will have your TIs make these slides available to you later in La Lima, so you can look for these. But obviously, you know, these are all slides from the different lectures, so you technically have all these slides already, but you'll get a copy of this later today. Let's go ahead and go to the chat to answer any questions that you have, okay? All right. All right, so there's a question about the logistics. Why is it that that in the multiple choice, I only let you move forward and not back? Very simply, it's to discourage cheating, okay? Because you can imagine with 200 of you all logging on from different places like home or somewhere else on campus, right? If you could go back and forth, then there's nothing to stop students from calling each other and comparing answers, right? Or working together on the exam. And I know most of you would say, well, you wouldn't do that, right? Because you're honest students. But at the same time, would you feel comfortable knowing that some of your classmates might be cheating like that? And if they get 100 on the exam and you get an 80, their 100 still counts as much as your 80 does, right? So it's really to discourage cheating. All of you are in the same boat though, right? Meaning, right, you all have to do it this way, right? Okay. So that's why so in the question, you say normally on a paper exam, you go back and double check your responses. Yes, unfortunately, you will not be able to do that, right? So it all comes down to how prepared you are. Because if you are prepared, right, and you take your time and you click on the right answer, the answer that you intend to be the right answer, right, then, you know, you don't need to go back and double check, right? Okay. Yes, a number of you are saying that, yeah, that's to prevent cheating, right? Okay, let's scroll down. Can I clarify the 5' to 3'? I assume you mean the orientation of a DNA strand. Remember, it all has to do with the very last nucleotide on either end, right? Is its 5' carbon the thing that ends up sort of sticking out there, or is it the 3' carbon with need hydroxyl group, the last thing sticking out there? Right, so that's the 5' to 3' nature. Will we need to be able to draw anything? No, you do not need to draw anything for the in-person exam unless you want to. In other words, if you're trying to describe something and you feel like a little illustration helps, you can. But the amino acids, you will need to know by sight. Okay, so you don't have to draw the amino acids, but you do have to study and review the 20 amino acids because you are responsible for those. All right, just like you were on the quizzes and I think on homework. What's the best recommendation for strategy for reviewing? I would say, you know, you have all the YouTube recorded lectures. You know, I only hold you responsible for stuff that I've gone over with you in lecture. The study guides, right, are my way of sort of bullet pointing those, right? So the stuff on the study guide is all fair game. I would not bother putting something on a study guide unless I felt it was important, right? Because if I didn't think it was important, I wouldn't just put it on there to confuse everybody, right? So use the study guides. I'm always amazed every semester at the number of students who don't look at the study guides, and then they ask me for like why they failed the exam, right? And they want like better study strategies. And I say, well, how do you use the study guides? And they say, oh, I didn't. Well, there you go, right? The study guides tell you how you should be studying. Okay, so it's up to you to actually use it. One thing you can do is that I give them to you in Microsoft Word format so that you can actually put spaces between the different bullet points or whatever. The best students, the ones who totally ace this class, they actually use those study guides as sort of optional homeworks, right? So for each question or bullet point that I put on the study guide, right, if you take the time to really try to type out like a 1-sentence or 2-sentence response to it, that forces you to confront whether or not you really know something that you might just sort of think you know, right? Because if you cannot write something in a response on when you're studying using the study guide, then when that same thing shows up in a real test or exam situation, right, it's not like suddenly you're going to know what to write then, right? So use that. It takes time, of course, but that's a really good strategy. And like I said, every semester, the students who ace the class, when I ask them how they use the study guide, that's what they do, okay? And then, yeah, so study guide, the lecture slides, the YouTube videos, use the book as sort of a additional way of supplementing many of the concepts, right? I would not study by trying to read the book first, right? That is a bad strategy because there's a lot of stuff in the book that we don't cover. So if you start from the book first and use up all your energy and patience trying to understand like the entire chapter, right, you're wasting your time and effort when it could be spent focusing on the things that you need to study, right? And I tell you what you need to study in the study guides and in the lectures, okay? Do we need to memorize all the structures of the amino acids for the test? Yes, you do, right? So you might have typed that question before. I already said that. Can I review secondary versus tertiary structures of proteins again, right? So go back to the slide. I can go back right now since we're here. So the primary structure of a protein is just the amino acid sequence, right? I think everybody's comfortable with that. Secondary has to do with the fact that if you ignore all the side chains, right? So in these 3 amino acid residues, the side chains are in sort of the darker highlighted rectangles. Just ignore those. What are you left with? You're left with the central part of the amino acid, right? That is the same for all amino acids. So secondary structure has to do with what sorts of angles and shapes form just because you have a chain of that central polypeptide backbone. So these illustrations in the middle, if you look at them carefully, right, identify if you can see the central structure of the amino acids in there and realize that no R group side chains are included in these illustrations in the middle of the slide, right? So what results is just interactions like hydrogen bonding that influence just the tendency of a polypeptide chain to fold up. That's your secondary structure. Now, if you take into consideration that all of these amino acids also have different side chains hanging off of them, right, then it gets even more complicated. That's why in the tertiary structure, right, it's a combination. These curvy helical structures are primarily secondary structures, right? You can see them. Or these sort of simple loop regions. Many of these are just a function of the polypeptide chain, but the overall irregular shape of this big, long polypeptide chain is due to all the R groups once you sort of factor them in, right? And that's why it's so important that we have 20 amino acids to choose from, and depending upon how you chain them together, the particular sequence, that will influence what overall 3-dimensional shapes, the tertiary structure, you can expect of this protein, right? Okay, hopefully that answered your question. Okay, So I think that's it for the chat. So good luck with your studying. If you have questions on the material, ask your TI first, because they are your front line for getting like the quickest response. And that way, they can address particular questions that are coming up with you and your classmates in your section. And that way I don't have to spend my whole day answering the same question from like 20 students over and over and over again. Right. But if there's anything that your TI cannot provide you a satisfactory response for, by all means, shoot me an email. Right. And I'm happy to then respond to you. Okay. But in order to keep my ability to respond efficiently to all the questions, that's why it's important that many times your question can just be answered by your TA first. All right. Okay. All right. Good luck with your studying. And don't forget to log in on Wednesday at 10:30 on La Lima to take your multiple choice part. All right. Okay. Bye-bye.